To give this talk. So our quick roadmap for the talk today. First, I'm going to go through an introduction to the biology of cancer, and then later Tom will talk more about cancer in the clinic in the second half of our talk. So let's get started with the introduction to the biology of cancer. So cancer impacts nearly all of our lives, and for 1 in 2 men and 1 in 3 women, they will be directly impacted by cancer and develop the disease at some point in their life. The rest of us will be indirectly impacted by cancer, as these 1 in 2 men and 1 in 3 women will be our close family members, our friends, colleagues, and our acquaintances. So probably almost everyone in this room has either known someone who has had cancer or maybe has had cancer themselves. And this year, 1.6 million people will be diagnosed with cancer, and cancer is estimated to kill 589,000 people this year. Now, if these numbers sound big or surprising to you, that's actually because they are. So cancer is the second leading cause of death in the United States. It kills almost as many people as heart disease and 4 times more people than the third leading cause of death. In those under 85, cancer is actually already our #1 killer. And so whether or not these statistics are new and surprising to you, or whether maybe you've heard them before or quite familiar with them, what we think is important to take away here is that cancer impacts a lot of lives, and the ability for scientists and researchers and clinicians to understand exactly what cancer is and then to develop therapies and treatments for cancer has the potential to impact not only the millions of people who have cancer right now in the United States, but also all of their family and friends. And so this is why Tom and I are pretty excited to give this talk to you today and share what we know about cancer. So what exactly is cancer? So cancer is characterized by the uncontrolled growth of abnormal cells. And asking the question, what is cancer, and giving this single definition here of uncontrolled growth of abnormal cells actually makes cancer sound like it's a simple single disease. But what we should keep in mind is that in our bodies we have over 37 trillion cells, and most of these cells aren't the same. So for example, neurons are quite different from blood cells, which are different from intestinal cells. Not only do these cells look different from one another, they're located in different parts of the body and they perform very specific functions. And so cancer arising in each of these different kinds of cells in these different parts of the body aren't the same. Furthermore, if we even take cancers that arise in the same organ, so for example lung cancer in the individual here on the left is going to be different from lung cancer in the individual on the right. These cancers may look different, progress differently, and respond differently to treatments. We also know that smoking can cause lung cancer, and so lung cancer that's caused by smoking is going to be different from lung cancers that arise in non-smokers. And so really, cancer is more accurately a term that refers to a collection of diseases, and not just a single disease. And this collection of diseases has in common the uncontrolled growth of abnormal cells. Now, the uncontrolled growth of abnormal cells can lead to the formation of a tumor, and tumors are a mass of tissue that lack normal function, and they cause disease by interfering with the normal function of the neighboring tissue. So we have 2 major classes of tumors. The first is benign, and so these tumors are localized and lack the capacity to spread throughout the body. So essentially they cannot metastasize. Our second class of tumors are malignant tumors, and these are ones that have the potential to invade nearby tissues and also spread to other parts of the body. And so when we're talking about cancer, we're actually talking about malignant tumors and not benign tumors. So we know that cancer cells and normal cells are different, and I wanted to take a moment to go through some of the differences between normal cells and cancer cells, since we're giving this definition of uncontrolled growth of an abnormal cell. So we have normal cells on the left and cancer cells on the right. So normal cells grow and divide in an orderly fashion, whereas cancer cells grow and divide uncontrollably. And so this is probably the most defining characteristic between these 2 different types of cells. And instances in which we want normal cells to grow and divide is, for example, during development, so when an organism is getting bigger, or if we get a wound. So for example, if we get a cut on our arm, we want the cells to grow just enough to fill in the cut, but not anymore. Cancer cells, on the other hand, will just keep growing even though they're not supposed to. Normal cells also die when they become old or damaged, so we don't have abnormal cells in our bodies, but cancer cells resist death and will continue to grow. Normal cells also carry out specialized functions in our bodies, but cancer cells typically lose the ability to carry out these specialized functions. And lastly, normal cells stick together in the right place in the body. So for example, we don't have intestinal cells in our mouths or lung cells on our skin, but cancer cells have the potential to invade other tissues and spread throughout our bodies. And so of all these characteristics, the most important one is this first one in relation to growth. So are there any questions about what cancer is that I can answer for you guys? Yes?
Are those differences always consistent, or could you have like cancer cells that are growing uncontrollably that haven't lost their original function?
I can't give you a specific example that I know of, but these are general characteristics that describe normal cells and cancer cells, and it's not absolute, so they don't need to have all these characteristics at one time. But these are, on average, in general, most of these characteristics are present in a cancer cell versus a normal cell. Okay, so I'm going to move on to talk about what causes cancer. So cancer is primarily caused by an accumulation of genetic mutations, and these mutations lead to changes in the production or activity level of proteins. And so in our first talk, we just learned that DNA encodes the information to make proteins, which are the molecular machines of our cell that carry out a lot of different functions. Now, if we have a mutation in our DNA, which I've depicted here in red, that mutation can potentially cause a change in the production level of proteins. So we might make too much of this protein or not enough, or it can actually change the protein itself. So since we say that mutations in our genes cause cancer, we often also say cancer is a genetic disease. And so ways in which we can get mutations is, one, we can acquire mutations. And acquired mutations are mutations that are the result of damages to the DNA in our cell that we acquire throughout our lifetime and are not heritable. So one way in which we can acquire mutations is through exposure to different environmental factors. And so a lot of us might be familiar with some of these. So one environmental factor is exposure to chemical carcinogens. So we know that smoking causes lung cancer, and the smoke that we get from cigarettes has a lot of carcinogens in it. We also know that asbestos, which is used in installation in old housing, can cause cancer. And then some workplace exposures, including formaldehyde, can cause cancer. Additionally, the pollution from coal burning power plants, as well as coal tar and coal tar pitch, can cause cancer. We can also get cancer from exposure to radiation. So UV radiation from the sun or from tanning beds can cause cancer, and this is why we're advised to wear sunblock and to avoid using tanning beds. And also exposure to X-rays and gamma rays can cause cancer. We can also acquire mutations through errors in DNA replication. So we learned in our first talk we have about 3 billion base pairs of DNA in our cell. So every time the cell wants to grow and divide, we need to make a second copy of this DNA so that each cell has the exact same copy of DNA in it. So making this copy of DNA of the 3 billion base pairs is equivalent to typing 60 words per minute, 8 hours a day for 50 years. And so that's a lot. And occasionally, sometimes a mistake is made. Just as when we're typing a document, sometimes we make mistakes. And this mistake can be passed on to the next cell. Now, luckily, our cells have machinery which help repair these kinds of mistakes. But as we know, spell check isn't perfect, and the machinery in our cells also isn't perfect. So we end up with about 1 mutation per 10 billion base pairs that are replicated. And so these are mutations that we can't do much to avoid, and they just occur naturally over time. Exposure to chemical carcinogens and radiation we can to some extent control or avoid. So we can decide whether or not we want to smoke, and we can decide if we want to put on sunblock or use a tanning bed. But these mutations, there's nothing we can do to avoid. And that explains why cancer also becomes more common as we age. Lastly, we can also inherit mutations from our parents. And so inherited mutations are just mutations that are present in our parents that get passed on to us. And these mutations are present in every cell of our body. So we have about 20,000 protein coding genes in our DNA, but mutations in most of these genes will not cause cancer. And cancer is primarily caused by mutations in 2 classes of genes, which are oncogenes and tumor suppressor genes. So first, I'm going to go through an analogy to help us define what an oncogene and tumor suppressor gene are, and then I'm going to talk a little bit more about what oncogenes and tumor suppressor genes are doing in biology. So our analogy is that cells must control their growth, like cars must control their speed. So oncogenes are genes that drive cancer when they're inappropriately turned on or activated, much like how a stuck gas pedal accelerates a car. So if we have a car and the gas pedal is stuck, then it's driving uncontrollably and crashes into the wall. If we have tumor suppressor genes, so these are genes that drive cancer when they're inappropriately turned off or inactivated, much like how a broken brake pedal cannot slow down a car. So if we have the same car and it's driving and we can't turn on the brakes, it's also driving uncontrollably and crashes into the wall. So essentially we get the same result here in this analogy, a car that's driving uncontrollably. But this result occurs through two different mechanisms. One is being on or stuck gas pedal, and the other is being off or a broken brake pedal. So in our cells, oncogenes are basically genes that are cancer related. So onco is the prefix for cancer or related to tumor. So these are like cancer genes or tumor genes. And in normal cells, these genes help cells grow appropriately when they're turned on at the right times. However, in cancer cells, there are mutations which cause this gene to always be turned on or activated. Tumor suppressor genes are genes which are suppressing tumors. So these are genes which help prevent cell growth or repair DNA damage. In cancer cells, these genes have mutations which cause the gene to be permanently turned off or inactivated. So essentially in cancer cells, oncogenes are always on and signaling for the cell to grow, and tumor suppressor genes are turned off. So the cell has difficulty preventing growth and repairing DNA damage. Now, an example oncogene is RAS, and RAS is a protein that transmits the signal to grow, and it was one of the first oncogenes that we ever discovered. So in our normal cells, we have the signal to grow here in purple, and when that signal reaches our cells, it eventually turns on a protein, RAS, here, and RAS transmits the signal to grow so that the cell eventually turns on all the genes that we need in order to grow and divide. And so in normal cells, a signal leads to growth, and so the cell only grows when it's supposed to. However, if we have a cancer cell in which we have a mutation in RAS such that RAS is always on, then essentially the signal to grow is always on in these cells, despite not having received the signal to grow yet. An example tumor suppressor gene is Bracha, and Bracha is a gene that helps us repair DNA damage. So if in our cells we have regular DNA and it sustains some sort of mutation which causes the DNA to break into two pieces, Bracha is a protein that helps the cell glue these two pieces of DNA back together so that our DNA is normal and our cells are normal. However, if Bracha is mutated, the cell can have a hard time gluing these two pieces of DNA back together, and so our DNA is mutated. And we can also get additional mutations in other genes that we may need that can drive cancer. So some of you may be familiar with Bracha already, as this gene is associated with breast cancer. So individuals with mutations in Bracha are at a much higher risk of developing breast cancer. So we can see here in purple that individuals with a Bracha mutation have a much higher risk of developing breast cancer than the general population, and ovarian cancer as well. And so a few years ago in the media, Angelina Jolie was talked about. She had a Bracha mutation and she elected to have a double mastectomy, a preventative double mastectomy. So some of you may be familiar with this gene already. So I'm happy to take any questions on oncogenes and tumor suppressor genes and mutations at this time. Yes?
Does Bracha affect more than just breast cancer?
So Bracha also affects ovarian cancer, so you have a higher risk of developing ovarian cancer as well.
So those are two female-related cancers.
It does slightly increase breast cancer in men as well, but breast cancer in general just isn't as common in men as it is in women. Okay, so I'm going to go through the last part of my talk here. So one question that may arise is a single mutation enough to cause cancer? It sounds like it's pretty easy for us to get mutations, and the answer is no. First, we're going to need mutations in the right kinds of genes, oncogenes and tumor suppressor genes. And cancer-causing genes also build upon one another. So here we have a normal cell here. And so, for example, over time it acquires a mutation either through exposure to a carcinogen or radiation or an error in DNA replication. And so this cell seems normal but is maybe more susceptible to developing cancer. Over time, another mutation is acquired, and this cell starts to become a little bit less normal. Another mutation may cause the cell to grow faster and look more abnormal, and eventually enough mutations are acquired such that the cell is growing uncontrollably and is very abnormal. And so this cell essentially is a cancer cell. And so in the average cancer, there are 20 to 80 mutations on average. And one of the things that we're working on is trying to figure out which of these mutations are actually causing cancer. And so, oh, sorry, I skipped a slide here. So individuals with inherited genetic mutations are actually more susceptible to developing cancer. And that's because individuals with inherited mutations essentially start out here in every cell of their body. So they're already more susceptible. Now, as I was saying a second ago, the average cancer has 20 to 80 mutations. And so one of the things we're trying to do is understand how these mutations are causing cancer and which of these mutations are actually important. So one of the things that we're using to do this is researchers are using cancer genome sequencing to help understand cancer. And some of our goals are to identify driver mutations as well as weed out passenger mutations. And so what this means is we're looking for the mutations that are really driving or causing cancer. And we're trying to weed those out, weed out the passenger mutations, which are mutations the cells have acquired over time simply because they're abnormal and growing rapidly. And these mutations may not be directly contributing to cancer. Another goal of cancer genome sequencing is to advance personalized medicine. And so Tom will come back to this a little bit more during his portion of the talk. So some of the things that we found from cancer genome sequencing studies is that certain genes are recurrently mutated in particular cancers. We've also found, through an analysis of almost 5,000 samples and 21 different tumor types, 33 new genes that we didn't know were involved with cancer before. And we also know that we still have a lot more genes to find. So what this information is getting us is that it's telling us a lot more that we didn't know about cancer biology. And it's also providing a number of potential new targets for cancer research and therapy, which are active areas of investigation in labs around the world today. So to summarize my portion of the talk, cancer is a collection of diseases that are characterized by uncontrolled growth of abnormal cells. And cancer is a genetic disease that's caused by acquired or inherited mutations. Mutations that cause cancer occur in oncogenes and tumor suppressor genes, and typically more than one mutation is necessary before cancer develops, and this also means that each cancer is unique. Lastly, cancer genome sequencing is being used to identify large numbers of potential new targets for cancer research and therapy. And I am happy to take any additional questions you guys have before I hand it over to Tom. Yes?
So how do you separate driver from passenger?
So that's hard. One way that I don't do cancer genome sequencing myself, and I'm not a bioinformatician. So some of it is that we take some of these genes and we actually do experiments in the lab at our bench to see if this causes different phenotypes. So we see if perturbing some of these genes maybe causes cells to grow faster, and that lets us know that maybe this gene is involved. And there's a lot of statistics that can go into it to help us identify which genes are significantly mutated and maybe drivers as opposed to not. Yeah. Yes?
Does one cancer-associated mutation make other cancer-associated mutations more likely to happen?
Some do. So if you have a mutation in, for example, like a DNA repair gene, so that means that you actually can't repair other mutations, so you are more likely to get mutations in your tumor suppressor genes or your oncogenes. Yes?
So the previous cancer genome sequencing study that you showed, is that like one particular study or is that in general in the field?
There are a lot of different studies being conducted by a lot of different researchers and a lot of different groups. The number for the 5,000 was from the Cancer Genome Atlas study. Okay. Well, thank you guys.
Can everyone hear me okay? Good. Okay, so I'm going to talk more about cancer from a clinical perspective, but first I just want to remind you guys I'm a medical student, I am not a medical doctor, and when I talk about treatments, this is general and for educational purposes, and I am in no way telling anyone that this is the treatment that you or your friends or family should get. Okay. So first I'd like to discuss how does cancer harm the body. And as Brittany told you, a tumor is a mass of tissue that lacks normal function. And additionally, tumors, whether they are benign or malignant, causes disease by interfering with the normal function of neighboring tissues. So when we think about this, the location of the tumor matters a whole lot. So a tumor in the brain, for example, is going to cause much more severe disease than a tumor on the skin, for example. Now there's one exception to mention, and that's blood cancers. So they don't form tumors because those cells are already circulating throughout the body. But they still cause disease in a very similar way, and that's by interfering with the normal function of your blood cells. Now, as Brittany talked about the difference between benign and malignant tumors can metastasize or spread to other parts of the body, and that allows them to cause disease in these new locations as well, which is generally why these are more severe tumors to have. Additionally, this makes treatment more difficult, as I'll talk about later in the talk. To illustrate this point, I'd like to talk about skin cancer. So all skin cancers account for about 3.5 million cases in the United States. And if you notice, this is actually higher than the number that Brittany told you at the beginning of the talk, because most skin cancers are very, very low risk and aren't actually reported. And so these aren't generally even considered when we're talking about the total number of cancers. The particular type of skin cancer which is counted because it's very dangerous is melanoma. So melanoma accounts for only about 2% of all skin cancers, but it accounts for about over 80% of all skin cancer deaths. And the main reason that this is so much more dangerous is because melanoma metastasizes very aggressively, whereas non-melanoma skin cancers are very unlikely to metastasize. Melanoma metastasizes early, and it metastasizes to dangerous places of the body, like the brain and the lungs. So again, metastasis is the definition, the potential for metastasis is the definition of malignancy. But luckily for all of us, metastasis is a difficult and rare process. So if we start with our melanoma skin cancer that is trying to get to the brain, one of these cells needs to learn a number of new tricks to be able to get from its original location to the brain. So first, it needs a pipeline, and this is usually either the blood or the lymphatic system. So the lymphatic system is one way in which white blood cells get around through your body to try to fight off infections. So first, the tumor needs to grow and invade large enough that it gets itself to a pipe. Once it gets there, a cell needs to break off and get into the circulation. Once in the circulation, it has to avoid your white blood cells, which are actually really good at detecting and killing off cancerous cells. It then needs to get to its new location, and think about this as a highway with cars rushing by. So it needs to get to the new location and stop, get out of the pipe, survive in the new environment and by itself without all of its friends, and then it needs to grow and proliferate into a new tumor. So very fortunately, metastasis is a rare event, and it's actually possible to have thousands or tens of thousands of cancerous cells circulating in the blood system, and not a single one of them will be able to survive in a new location. So if a metastatic tumor hasn't grown large enough to the point where it's causing disease, the main way that we detect it is through imaging. So this is a CT scan, and what you're looking at is if I had been cut in this plane here, and you had a bird's eye view looking down at me, and what is left there. So this is the patient's heart in the middle, and the black areas are the patient's lungs. And so they're black because they're usually filled with air. Unfortunately, all of these small circles are metastatic disease, and this is actually from prostate cancer. So you got to think that each one of these metastatic tumors originally started as one cell. And this one cell divided into 2, which divided into 4, which divided into 8, and so forth. So to have enough cells to form a tumor that we can see on imaging, you need usually at least 1 million cells. And this takes 20 of those divisions. So that actually takes quite a long time. But unfortunately, once you have 1 million cells, if that continues to divide, each cell divides into 2, it doesn't take that many more divisions to have a much bigger tumor. So only another 5 divisions off of that, you've got a tumor which is 30 times the size in 3 dimensions as that initial tumor. And with another 5 divisions from that, you've got a tumor which is 1,000 times the size of the smallest one that we can detect. Unfortunately, what this means is that this metastatic cell spent 2/3 of its lifetime as a tumor too small for us to be able to detect. And if in a patient we already have a metastatic tumor that we can detect that's in this range, unfortunately, chances are that there's another cell somewhere else in the body which is hiding out in this 1 to 20 division range, which is why metastatic disease is one of the worst factors when we're talking about cancer staging. So staging is the process of categorizing cancers by severity. And staging guides our treatment decisions, the kind of prognosis that we can give a patient, so we tell them what to expect from their disease, as well as clinical trials and research. Things that we consider when we talk about staging are the size of the original tumor, whether there's lymph node involvement, so again, the lymphatic system is one way that cells can get through the body, and think of lymph nodes as stations. And we look at the lymph node which is closest to the tumor, and we see, are there any tumor cells in that lymph node? If there are, that suggests that cells have started moving away from the original tumor, but this is their first stop in the process. Whereas if we already see distant metastases somewhere else in the body, that means the cell has already traveled a long distance and started to grow a new tumor. That's a more serious factor to see. Physicians call this the TNM staging system, but you're probably more familiar with the individual staging systems for different cancers, which usually range from 1 to 4. So to come back to melanoma, whatever stage a patient is put into is highly predictive of how long they should survive from the disease. So the y-axis here is the probability of survival, and the x-axis is over time. And you can see that patients who are in with stage 1 disease do pretty well. So stage 1 is going to be a localized melanoma that's not very deep, and it hasn't gone anywhere else in the body. Stage 2 disease is still a localized tumor. We haven't found cells anywhere else, but it's going to be bigger or deeper into the skin. Stage 3 already shows lymph node involvement, and stage 4 shows distant metastases somewhere else in the body. So this really emphasizes the importance of catching cancer early, because if we can still catch the disease, even a very dangerous one like melanoma, at an early stage, the patients still do quite well. So to summarize this first part of my section, tumors cause disease by interfering with the function of normal tissues around them. Malignant tumors can metastasize, but just because a tumor has the potential to metastasize, it doesn't mean it already has. Staging allows us to categorize cancers by severity, and the presence of metastases indicates the highest stages. And I'd like to take any questions.
You say the tumor interferes with neighboring tissue. Is it just because of the physical presence, or is there like chemical interactions?
Yeah, so the physical presence is a big thing, particularly as you're talking about larger tumors will have more effects. But there's a variety of different ways in which tumors can cause disease, and this is kind of a simplified model. They can release chemicals, they can release hormones, which have all kinds of impacts across the body. So it is a little more complicated than that.
On your 4-stage graph on the previous slide, is that with or without intervention and treatment?
So it's a good question. It's unfortunately about the same no matter what. So that's the prognosis going up front. So yes, that would be with treatment, because if you were stage 1 without treatment, you'll bump down to those other stages.
Can you have the metastases make their own metastases, or does it always have to start from the original tumor?
Yeah, that's a good question. So metastatic tumors can metastasize again, but generally if you're already at the point where you have a metastatic tumor which is big enough, the disease which you're likely to be dealing with is going to also have originated from that first tumor.
Are the blood cancers, by virtue of definition, because they're already in the circulation, are they already a metastatic cancer, or are they like, do they occur in stages like the solid tumors?
Yeah, it's a good point. So there are stages for blood cancers. You can definitely argue that by definition they are metastatic, but they behave a little bit differently because they're not going to form new tumors in other places. So it's not such a big deal that they're metastatic, and they are going to be causing more or less the same disease just by their nature of being in the blood.
People sometimes talk about the RAS pathway having to be related to growth abnormalities and P52 and Bracha being related to mutation rates increasing in cancer.
Sure.
Can you say anything about the pathways or genetic mutations that are related to metastasis?
Yeah, so there are lots of different pathways related to metastases, and that goes back to sort of all of those different steps which I had talked about. So for example, in order to get into the pipe, a lot of cancer cells will start releasing an enzyme to degrade the wall of the pipe. So expressing that is one way. Matrix metalloproteinases is one way in which you can have mutations and changes in expression. Another one is how do they stick to the wall so they can stop and then get out. And so there are particular receptors and molecules on the surface of the cell that they can start expressing and have mutations in. So there's a whole host of mutations that classify for metastasis.
Would you expect some of these changes to activate the immune system?
Yes. So right. So cancer immunotherapy is an emerging field. I'm not going to talk too much about it today, but the more different the cancerous cells are from the normal cells, the more likely they are to be detected by the immune system.
So are there different types of cancers that would give an inflammatory or immune response signal much earlier?
Yeah, so there's a lot of research into this field at this point, and we're trying to figure out ways to try to help the immune system to combat cancer. So I'm going to move on at this point just to try to keep on time, but I'm happy to talk with anyone else at lunch or during the breaks. So for the rest of this talk, I'm going to talk about treatments. And so our three major current types of treatment are surgery, radiation, and chemotherapy. So surgery is very effective if we have a localized single tumor to work with, because it's usually possible to just remove the entire cancer and then rid the patient of disease. Some considerations when we think about surgery are, where is the tumor? Can we get to it safely through surgery? How big is a tumor? Can we remove it without severely damaging the tissues around it? And we also consider, can we remove margins, which are areas of normal tissue around the tumor that ideally we can remove to make sure that we get all of the cancerous cells. Because if we leave behind even one cell, that can continue to grow and form a recurrent tumor. Radiation therapy is also helpful for localized treatment and particularly helpful for cancers that we can't get to surgically. So again, this is a CT scan in the same plane. This is the patient's heart, and the black area is the patient's lungs, and the white mass here is the patient's tumor. The different circles indicate how much radiation we're going to deliver to that tumor. The goal here is to deliver as much radiation as possible to the tumor and as little radiation as possible to the surrounding areas. And the main way that this is done is by delivering radiation from a number of different angles so that it overlaps on the tumor, but the amount through any given surrounding tissue is limited. There are other considerations that go into this, such as different parts of the body are more or less sensitive to radiation. So this beam would go through the patient's spinal cord, for example, which is very sensitive. So you'd skip that beam. The heart can take some radiation, but less than, say, muscle and skin. So there's lots of calculations which goes into a very specific radiation treatment plan to deliver as much radiation as possible to the tumor and as little radiation to the areas around it. Also remember, I'm only showing you a two-dimensional slice here, and we can actually use beams of radiation in 3 dimensions as well. Now, Brittany had told you at the beginning of the talk that radiation is something that causes cancer, and that's absolutely true. At a modest amount, radiation will damage DNA, but not to the point that it kills the cell with that DNA. No cells can survive particularly high doses of radiation. But one of the long-term potential side effects of radiation therapy is a somewhat increased risk of other cancers in the tissues surrounding the area that had been irradiated. But almost universally, the risk of this initial tumor that you need to work with is much higher than this somewhat increased risk long-term for the patient. Now, chemotherapy is a use of drugs to treat cancer, and this is really our major treatment for when cells are in different parts of the body. Now, to think about this, we need to develop drugs which kill cancerous cells but don't affect the normal human cells in the body. And as an analogy, I like to think about antibiotics. Now, the goal there is to develop drugs which kill bacteria without killing the normal human cells. Now, this is actually the size difference between E. coli and a normal human cell, and there's also 3 billion years of evolution between these two different types of cells. So there are natural differences in biology to design these drugs to target the bacteria. There are challenges, and you'll hear about those later today. But overall, that's easier to do than developing a drug which is specific for the tumor cell, but leaves the normal cells unharmed. The main way that we've done this for decades is to develop drugs that just generally target rapidly dividing cells, because this is the definition of cancer. As you had seen earlier, every time a cell needs to divide, it makes an entirely new copy of its DNA. So we have a variety of drugs which prevent that from happening. And there's also a particular set of mechanisms which need to occur in order for one cell to divide into 2. So we also have drugs which inhibit that. Unfortunately, this leads to a number of predictable side effects, which are damages to the other rapidly dividing cells in the body, such as your hair, your skin, the cells lining your stomach and intestines. This is where the nausea and loss of appetite comes from, as well as the cells in the bone marrow. So this is where the fatigue and the susceptibility to infection comes from. So one of our goals going forward, which I'll talk about more, is developing drugs which are more specific for the cancers, which can be helpful in killing the cancers while leaving your normal cells more untouched. One more thing to consider is that cancer cells can become resistant to chemotherapy. Now, tumors are made up of a diverse group of cells. They all have different mutations, so they have shared mutations, and they additionally have new mutations. These cells are always gaining more mutations. Chemotherapy, particularly the ones which harm DNA, can unfortunately also cause more mutations if it's not at a dose enough to kill the cells. And you only need one cell to develop resistance, which can then form a new tumor. So one of the ways that we combat this is to try to always target, when we can, with more than one drug. So if there's a 1 in a million chance of a cell becoming resistant to drug A and a 1 in a million chance of cell becoming resistant to drug B, if we're treating with both drugs A and B, there's a 1 in a trillion chance of a cell developing the mutations necessary to be resistant to both drugs at the same time. So whenever possible, we treat with multiple drugs, at least 2, and often more than that. So to come back to melanoma, whatever stage the patient is at is highly related to which treatment we decide to use. So if we're talking about stage 1, so again a very localized tumor, we can usually remove that with surgery and the patients will do quite well. If we're talking about stage 2, one way that we might treat that is to remove what we can with surgery and then irradiate the area to make sure that there wasn't anything we left behind. Once we get to the point that there are cells in the lymph node or other places in the body, we'd likely still remove the primary tumor, potentially irradiate the area or irradiate the lymph nodes, and as well as to give chemotherapy to try to kill cells which have already gone to other places in the patient's body. Now, moving forward, as I had mentioned, we're very interested in developing targeted therapies. So this is using our increasing knowledge of the mutations in the biology driving cancer to develop more specific drugs. And our classic example of this is for a disease called chronic myelogenous leukemia, or CML. And in CML, there's one specific mutation that almost always drives the disease. So 2 chromosomes, chromosome 9 and 22, parts of their chromosomes break off. And instead of the cell gluing them back together where they're supposed to be, they switch the pieces that break off and then glue them back together on the wrong chromosome. And what happens there is that 2 genes, which are normally very far apart from each other, BCR and ABL, are fused together. And the BCR-ABL oncogene is what drives the cancer. So this gives an uncontrolled growth signal to these cells. Now, this new chromosome that forms is called the Philadelphia chromosome, and you can actually see it under the microscope. And this is why we were able to figure this out quite some time ago. More recently, we developed a drug called imatinib, which very specifically blocks BCL-ABL, and it's very effective at killing off CML cells with the mutation. But since the normal cells don't have BCR-ABL, these 2 genes are still separate, this drug has quite fewer side effects than our more typical chemotherapies. Just to look at how long this took, the Philadelphia chromosome was first seen in 1960. Imatinib was patented in 1992, and it wasn't until 2001 that the FDA approved the drug. Fortunately, we're living in a time where we know way more about cancer than we ever did in the past due to new technologies. So the Human Genome Project finished in 2003. We sequenced our first cancer genome in 2006, and by 2015, we've sequenced over 1,000 cancer genomes and parts of many, many more than that. So our knowledge of the changes in DNA, which are driving cancer, is much, much larger than it has ever been at any point in the past. To illustrate this, I'd like to come back to melanoma again. So it was found fairly recently that 50% of melanomas include a very specific mutation in the BRAF oncogene. And this is the same RAS pathway that Brittany had shown you earlier. So a growth signal comes in, activates RAS, which activates another protein, which is actually BRAF, which activates other proteins, which causes the cell to have grow and proliferation signals. However, in tumor cells, particularly in these melanomas, a mutated BRAF means that the signal is always on. The cell is always driving to grow, no matter what is happening upstream, and even if you have a normal RAS. So we've developed a couple of drugs, one of which is vemurafenib, which specifically inhibits mutated BRAF, but has very little effect on normal BRAF. So this is effective at turning off this signal in the melanomas, as well as actually killing these cells, because cancers often become addicted to these oncogenes. So even though you're not only turning off the signal, but these cells can't survive without that signal. And to come back to our timeline, the BRAF mutation we only found in 2002. The first inhibitor was approved in 2007, and a second inhibitor was approved in 2011. So our ability to develop and get these drugs into patients is much better than it has ever been before. And to further illustrate this, this is just a small list of targeted treatments which are helpful for a variety of different cancers, are approved for a variety of different cancers, targeting a number of different genes, and most of them have been approved within the last 15 years. And there's many, many more in the pipeline of development. So where we're headed is an era of personalized medicine. And the idea here is that we treat a patient's tumor based on the specific mutations causing that tumor. We don't need to lump tumors together as breast cancer or lung cancer. And the way to do this is to just take a biopsy of a tumor, take some of the patient's blood, and sequence both of them. Look to see what's different in the tumor compared to in the blood, because those mutations are likely to be what's driving that person's cancer. Then we can compare that data to those of many other patients. So we can see who else has had those mutations. What were they treated with? What worked and what didn't? So then we can decide on the most effective treatment for that particular patient based on their particular mutations. Ideally, there'd be a targeted therapy for those mutations, so we can also use this information to try to develop more targeted therapies, which will include more patients over time. So to summarize this last part, surgery is effective at removing single localized tumors. Radiation therapy also helps with localized tumors, particularly those which we can't get to surgically. Traditional chemotherapies target cells throughout the body, and targeted therapies are being inspired by our increasing knowledge of cancer biology. And just some take-home points for the entire talk. Cancer is a genetic disease caused by DNA mutations. Cancer progression is tied to more mutations. Metastasis is the spread of cancer to other tissues in the body. We use surgery, radiation, and chemotherapy now, and we're very interested in developing more targeted therapies to attack the underlying biology behind patients' cancers. So I'll take any questions.
So, vemurafenib, how effective has that turned out to be? I mean, this is 2015 now, and that was 2011.
Right. So it is effective, but it's also, so going back to resistance is really the real issue there. So so imatinib was only the first BCR-ABL inhibitor, and we've had many more come behind it, which are more effective and more helpful for patients which become resistant to imatinib. And we're not quite there yet with the drugs for the BRAF mutations. So while they can be effective initially, we need more drugs to kind of come behind them to really help out these patients.
And what about melanoma patients who don't have BRAF?
So unfortunately, it's still the same traditional treatments. So it's surgery, radiation, and traditional chemotherapies.
What are the genetic mechanisms to drug resistance?
So cancer cells are always picking up more mutations. So as we had mentioned before, you can have it's pretty frequent to have mutations in DNA repair genes. So the mutation rate in cancerous cells is high. So, and you're also talking about millions, hundreds of millions, billions of cells. So they're picking up mutations randomly. And if you're putting a stress on the whole population of cells, so if you're giving a drug and you kill most of the cells, that allows the one cell that's got a mutation that allowed it to avoid the drug to then survive and grow up into that niche which is left behind by the other cancers. When we talk about the targeted therapies, they tend to be mutations related to the same gene, so BRAF, for example, but there are also mutations where a cancer cell just learns to export drugs. So the drug comes in, the cancer cell kicks it out, and that is something which is very challenging for us.
So you mentioned before with chemotherapy treatments now that they have adverse effects on rapidly dividing cells in our body now. Mm-hmm. Are there other therapies that exist out there that essentially arm our own immune system to specifically target tumor cells or metastases?
There are, and they're mostly recent developments, but it's an area of intense research, and I wasn't able to really fit it into this talk. But there were just a couple of drugs published in the New England Journal last week that is actually effective for melanoma, so late-stage melanoma, which works by a very similar mechanism of using your own immune system to try to kill off those cells.
Usually cancer requires a bunch of mutations, so like there could be cells with only a few of those mutations that haven't yet gone nuts. Does that mean that there could be a whole lot of sort of precancerous cells throughout the body that are a few steps away from becoming cancer? So we just like have all cancer all the time?
Yeah, so that's one of the challenges, is there are always cells which are somewhere on that spectrum. And, it's hard to know where they are, and this is where a lot of our screening tests come in. So, for example, colonoscopies are about trying to find those cells that are on that spectrum before they get to the point that they're cancerous. So that's, another area of research. I think I'm getting the signal to move on, but I'm happy to talk more at lunch.
Just a 5-minute break, and Tom and Brittany can stick around to answer questions during that time. But yeah, take a break.
Sure.
Hello. For either of you.
Oh, good.
Actually, the first question is that, in general, I wonder whether